Welcome to the Apron Academy. This video was specially designed for the dietitian in training, also known as the RD to be. Today we're talking about vitamin B12. So this one is pretty popular. It's a kind of a almost buzzword in our culture today. So first, what I want to talk about is where we can find it. So you can get it in meat, dairy products, seafood, fortified foods. You can't find it in any plants except for algae. And that's not really a popular plant to eat. So just to be mindful that if you are on a strict vegetarian or vegan diet, you may be low in vitamin B12. So, as always in many of my videos, I'm going to show you the chemistry of it. This was a quick little mock-up drawing that I made. So it looks a little messy, but it is this complex. You have a whole bunch of stuff going on here. But this altogether is vitamin B12. So, this structure is called Transcobalamine, and this cobalamine has a central cobalt atom surrounded by this heme-like Corin ring. So, I want to point out that there are many other structures that are similar to this. They may be slightly different, but that's going to be important going forward. Those structures are carotenoids and cobitamides. So just keep that in the back of your mind as we go forward. Before I move the page, I want to talk about, in most of our vitamin videos so far, we have talked about how for absorption, many of the large molecules need to be broken down or hydrolyzed into a smaller molecule so that it can be absorbed. That is not the case for vitamin B12. It always is going to stay in this large structure, and this is a ginormous structure. So, how is it going to be absorbed? So, I have my little dude here that I've had in the past, and we are going to imagine that our little dude here is eating some vitamin B12 in his meats or dairy or fortified foods. And so the B12 is traveling down into his stomach, and now I'm going to go into this image where I've kind of enlarged his stomach. So we have B12, and in the stomach we also have something called R binder. So I want to mention that B12 is released from proteins by pepsin, and the stomach acid that is produced by the parietal cells. So these little guys here, they're not just here, they go all around the lining of the stomach. But these are our parietal cells. These parietal cells, while I'm on the topic, they also produce hydrogen and this intrinsic factor. I'm labeling it IF. That is intrinsic factor, and it's not just 2. It releases multiple, but I've just written 2 in here. But that's produced by our parietal cells. So, intrinsic factor, you may have heard of this before. It is important for B12, But the intrinsic factor cannot bind to B12 in the stomach because it is so acidic in the stomach. So in order for B12 to travel through the stomach, it binds to this R binder. So it goes out, both the hydrogens and intrinsic factor and this R binder plus the B12 travel down the stomach and into the small intestine. So as it's in the small intestine, things are coming in, things like bile and pancreatic enzymes, and it changes the pH of our small intestines to be a little more basic. So in the small intestine, because it's more basic, this R binder is broken off of the B12, and the B12 is able to create a complex with the intrinsic factor. While I'm on this topic of the stomach, I want to talk about how this is an acidic environment. So the reason that B12 is able to move along is because of that acidic environment and able to bind to the R binder. So when you're on chronic antacids, things like Tums or anything, it lowers the pH in our stomach, and we do have to take into consideration that that may also affect our B12 absorption. So anyways, moving on, we have our B12 intrinsic factor complex, and it's going throughout the intestines, and we need to get into the body, so we have to go into this these cells, and this is my bigger version. So we have the B12 intrinsic factor complex, and in order to get in the cell, we have a receptor called cubulin. So this complex is able to travel through cubulin to get into the cell. This takes place by receptor-mediated endocytosis. That is how it gets in. So then in the cell, the B12 and the intrinsic factor lose their bond, and then B12 is able to go throughout the rest of the body into the blood and so forth. So as it enters the blood, it has two transport proteins that B12 has. So the first is Transcobalamine. This is our main one. When you think of B12, you want to think of this transport protein, Transcobalamine. So this binds to B12 and transports B12 to all other tissues. This also is completed through receptor-mediated endocytosis. So then we also have the second one, Heptachorin. And remember how at the beginning I mentioned that B12, that big old chemical structure, has similar looking things that aren't exactly B12. Well, this Heptachorin primarily transports those things. So it helps excrete the if I can say it, coronoids and cobinamides throughout, or helps excrete them from the body. Now we have two functions for B12. The first is in methionine synthase, and then the second is mitochondrial methyl malonyl, and goodness, I just can't talk tonight, Coamutase. So talking about the first one, let's talk about methionine synthase. So this might look familiar, and if it doesn't, I encourage you to go back and watch my video on folate, and it's towards the end when we talk about this methionine cycle. So kind of ignore this for now, but in this cycle, we have the 5-methyl tetrahydrofolate coming up, we can get it, we get it from the folate, but then we talked about in the video that we need to drop the methyl, give it to homocysteine, and then the homocysteine becomes methionine. We no longer have the methyl, so we just have the THF, and ta-da, there we go, able to go through the cycle. Well, I have this circled here because there's a little more to it than just handing it one to the other. Now, if you look here, this is the 5-methyl tetrahydrofolate, and then this is our methionine. So this is our goal. We need to get here. So if you follow these arrows, and I forgot to add, there we go. If you follow these arrows, then we can follow the 5-methyl tetrahydrofolate to this methylcobalamine, which is the B12 here, into making methionine. So there are a couple things we need to know with this. So this methionine synthase is important. We cannot run the reaction if we don't have the methionine synthase. It also requires this methylcobalamine as a cofactor to run the reaction. So basically what I'm trying to say here is, in order for us to have this methylation, we need B12, and we can see that right here. We cannot generate methionine without B12. Now, just like we talked about with folate, if we don't have enough folate, then we're going to have high homocysteine levels. Now, if we don't have enough B12, we also are going to have high homocysteine levels, and we talked about before how that increases cardiovascular disease risk, and we also fail to have this methylation, which could lead to cancer risks. Now, moving on to this second function, as a mitochondrial methylmalonyl CoA mutase. So this might look just a little confusing, but imagine, or just basically understand that these all go down. We have this L-methylmalonyl CoA. We need to get to succinyl CoA, which functions in the Krebs cycle, and we've talked about before that the Krebs cycle is needed for life. So in order to turn this guy into succinyl CoA, we need this going on. So we have the L-methylmalonyl CoA mutase. We need this 5-deoxyadenosylcobalamine, which is vitamin B12. It uses this 5-deoxy, basically vitamin B12, to successfully make the succinyl CoA. So, if there's no vitamin B12 in our bodies, we're going to have a buildup of this L-methylmalonyl CoA. And you notice that this is a one-way reaction, but with this, if there's buildup here, it's going to go back up the reaction. And what it then does is there's a buildup of methylmalonic acid. So we can look at this methylmalonic acid in our blood to see if we have a B12 deficiency. Now, we don't want to look at the homocysteine levels up here in our blood because these can be increased because of folate, and we'll see later that it can be increased because of vitamin B6. So know that if we don't have enough vitamin B12, we're going to have increase of this, which leads to an increase of methylmalonic acid. So now what else happens if we don't have enough? We're going to see a couple of primary manifestations. This happens because of a diet or defect. So we talked about a strict vegan diet, you're going to have lower B12, or you can also have a defect in absorption. There's a lot of stuff going on in absorption with the stomach and into the intestines. A lot can go wrong, so any little defect can affect the amount of B12 that we have. So these primary manifestations, what we would basically see is megaloblastic anemia. So we saw this also with folate. It's where we don't have enough red blood cells, or they don't mature. So we just have these large blasts and they don't have any function. So we're not able to synthesize purines, pyrimidines, or red blood cells with this anemia. With the neurological disease, we see demyelation of axons. And we also have malabsorption disorders. So here's this pernicious anemia. This is a lack of the intrinsic factor. It's an autoimmune destruction of our gastric parietal cells. So those cells along the stomach line. So we can eat all the vitamin B12 we want. And even if we have enough in our diet, we won't be able to absorb it because our gastric parietal cells are damaged. Then we also have atrophic gastritis, where our stomach acid is reduced. And we talked a little bit about that before. If we have antacids or something, where that just does not create the best environment for B12 to bind to the R binder and then travel through the stomach into the small intestine. So it takes a long time to detect a deficiency because vitamin B12 can be recycled as it goes through the kidneys and then back up into our body. So we talked a lot about B12 versus folate, and I do want to mention real quick about some blood tests, so how you can tell the difference between a folate versus a B12 deficiency. So if you have a low plasma vitamin B12, but a high methylmalonic acid, what do you think that deficiency would be? It would be a vitamin B12 deficiency. So now, if you have a normal plasma vitamin B12, but with a low plasma, and then you have red blood cells and folate, you have a folate deficiency. And throughout all this, you can see that B12 is vital to our life. If this video was helpful for you, please like and subscribe so that you can be the first informed about new upcoming videos. Thank you.